Bismillahirrahmanirrahim. Now, this is the second video on transport in humans. This is 5090, the new syllabus for 2023. And we're going to discuss 11.3 and 11.4, which are the syllabus points in this video. Now, make it a habit is to go through the syllabus before you start the chapter, and then when you revise the chapter, then go through the syllabus again and see if there's any points that you have missed. So the first point is name the main blood vessels. When it says name, that means you have to know the names. And the names of the blood vessels which you are supposed to identify is aorta, vena cava, pulmonary artery, pulmonary vein, hepatic artery, hepatic portal vein, renal artery, and renal vein. So you must be able to diagnose them and define them or describe them, not describe them so much as label them because it says name. And you must know which organs do they connect. Where do they go from? Where do they go to where? Like the pulmonary artery and the pulmonary vein. Hepatic is something to do with the liver. Hepatic artery, then renal is something to do with the kidneys. So you know which part of the body are they talking about. Then describe and identify on diagrams and photomicrographs the structure of arteries, veins, and capillaries, the relative thickness of the wall, the composition of the wall, which is made up of muscle and elastic tissue, the diameter of the lumen, and the presence of valves. Then explain how the structure of the arteries is related to the pressure of the blood that they transport. So pressure of the arteries, pressure of the veins, and pressure of the capillaries. So basically how the structure is adapted for its function. Then 11.4 is all about the structure of the blood, and it is identify the red and white blood cells as seen under the light microscope in diagrams and photomicrographs. You list the components of blood as red blood cell, white blood cells, platelets, and plasma. So we've got four stuff in it. The plasma is that liquid part, which I call the shorba, and of course the platelets and the white blood cells and the red blood cells. Then state the functions of the red blood cells: oxygen transport, white blood cells, antibody production, and engulfing pathogens by phagocytes. Then platelets, converting the soluble fibrinogen to insoluble fibrin to prevent blood loss from the entry of the entry of pathogens. Then plasma, what is the function of the plasma? And the fourth is describe the transfer of substance between blood and capillaries, tissue fluid, and body cells. First point is name the main blood vessels. So name means, of course, you must know, they will give you a diagram which is not labeled, and then you'll have to label it. So let's look at all of them. There are 9 that we have to know. So the first ones are the vena cavas. Let's talk about the vena cavas. The vena cavas are coming from the body. So there will be the superior vena cava, which is coming from the head, which is this one, and it's coming to the right atrium. Then the inferior vena cava is coming from here and it's coming into the right atrium. Then we come to the deoxygenated blood, which is the vena cavas. So both the vena cavas bring blood to the right atrium, right? Then it enters the right ventricle. Then we have the pulmonary artery. So this is the pulmonary artery which is arising from here and going here to the lungs. You can see these are the lungs here. And so the pulmonary artery goes here to the lungs. Of course, as I told you in a previous video, there'll be 2 pulmonary arteries, one going to both the lungs. Of course, this is a diagrammatic representation. So here it goes to the lungs. In the lungs, it is oxygenated, and then the pulmonary vein is going to return this, and there are going to be 4 pulmonary veins. This is what we discussed in video 1. So pulmonary vein will bring it back to the left atrium. Then the left atrium, left ventricle. And then, of course, we have the biggest artery coming out of the body, which is the aorta, which comes out here. And this has been labeled as the aorta. Now, the aorta comes down. And the arteries that you have to understand is the hepatic artery, which goes to the liver. So this is the liver. Then the renal artery, which goes to the kidneys. Mesenteric artery goes to the intestines. Yes, we don't have to know that. There's no name mentioned in the syllabus, but you have to know it that there would be an artery going to the intestines. But then there is no vein. I mean, if you look at it, there's a hepatic artery and there is a hepatic vein which goes and enters the inferior vena cava. Every organ has an artery and a vein. So hepatic artery enters the liver, divides into arteriole, capillaries, then venule, and then vein. And the hepatic vein is going to enter the vena cava, which is going to take the deoxygenated blood to the right atrium. Similarly, there'll be the renal artery which goes to the kidneys, and then there's going to be a renal vein. So there's going to be two renal veins, two kidneys, two renal veins, and they will enter into the inferior vena cava, which will enter and take the blood again to the right atrium. But then there's another one which is a connection between the intestines and the liver, and this is called the hepatic portal vein. Hepatic portal vein connects the intestines to the liver because all the food which is digested and absorbed enters the hepatic portal vein. Of course, there's only the fats which enter the lacteals and enter the lymphatics. So most of the food which we eat is going to be digested and is going to be taken to the liver through the hepatic portal vein. Now this is another diagram to make it further clear to everybody. What are we laboring and where are we laboring? So here you can see pulmonary arteries, pulmonary vein, hepatic artery, hepatic, they haven't shown you the hepatic artery, then the renal artery, then we have basically the inferior vena cava and the superior vena cava and the aorta here. So another diagram to show you the details, just to make you revise it and recap it. Basically what we have to understand, that every organ of the body has to have an artery which is carrying blood to that organ and has to have a vein which is carrying blood away from that organ. I said every organ of the body. Whether it's the liver, whether it's the lungs, whether it's the kidney, whether it's the stomach, there has to be an artery and a vein. So there has to be an artery and there has to be a vein. Now, the basic distribution is when an artery enters an organ, a kidney or a liver, it divides into an arteriole. And then there are capillaries. Capillaries are one cell thick and they will allow substances to pass in and out. Some things will pass in, some things will pass out of it. That is the only place where things are going to move in and out of this transport system. It's just like you are going in a car to your school, so if the doors don't open, how are you going to get off at school? Or how do the doors don't open, how you get into the car when you are going from your house? So capillaries will allow actually the function of the transport system to take place. Like you going from your house to your school, but if the doors don't open, you can't enter the car. If it doesn't open in the school, you can't get out of the car. So this is the place where the function of the transport system is going to take place, and then the capillaries will all join. So there is a network of capillaries all over your body. And then we have the venules, and then we have the veins. So this is the normal distribution that we have in every organ of the body. This is another diagram showing you this in detail, how the capillaries are the joining part between the arteries and the veins. Now the basic points of an artery are what? They have a thick outer wall. The wall is very, very thick. When you think of it, it's very thick. Why? Because it has to withstand a lot of pressure. Why? Because all arteries arise from the aorta. Aorta arises, you see, all arteries arise from the aorta. And the aorta arises from the left ventricle. And the wall of the left ventricle is thicker, so it exerts more force. So basically, the force is being generated by the left ventricle, not by the aorta. So the left ventricle wall is much thicker. I said the left ventricle wall is much thicker than the right ventricle, so it exerts more force. So if it exerts more force, that means the aorta carries blood at high pressure. So in order to withstand high pressure, this is the word that I use, withstand high pressure, there's a thick wall. Now in the veins, the pressure has been lost, so there's a thin outer wall. Then it has a small lumen. This has a large lumen, and it has valves in it. And then when you see it, what is left? If you look at it more carefully, what is left? It's just that, I'm going to draw it red, it's just that inner layer which is left in the capillary. This layer is only left now here, but this layer was in all the three. But outside the three, there was, of course, outside the inner lining, we call this the inner lining of the blood vessels, there is a muscle layer and there is elastic tissue in this wall here. There's smooth muscle and there is elastic tissue. But the vein wall is thinner and it has less muscle in it. Why? Because now the pressure is greatly decreased by the time the blood has reached the veins. Another diagram showing you this is the arteries have a very thick wall, and it is the middle layer which actually contains the muscles and the elastic tissue, and then there's an outer layer. So there are three layers: inner layer, middle layer, and outer layer. And of course, in the veins, we have valves. Why? Because especially in the leg veins, when blood is returning, when a person is standing up straight and blood is returning against gravity, well, there's a lot of chances that the blood, sort of is pulled back into your feet. But that doesn't happen. Allah Ta'ala has made you beautifully, and that does not happen. And that has made you the fact that all your blood, which is circulates all around your body, whether you're sleeping is a different story. Then the effect of gravity is finished. When you're standing up, gravity is pulling the blood down, so that blood has to return against gravity, and that's quite a difficult task. So this is why veins have valves. Normally, there's only in the arteries, there's only at the base of the pulmonary artery and the base of the aorta where there are valves in the heart only. In the veins, you have valves everywhere. And please remember, there are three flaps. They show you two flaps, but actually there are three flaps. One flap, two flap, and three flap. But because it's a cross section, so you haven't shown you the three flaps. At any given moment, about 30% of the blood in your systemic circulation will be found in the arteries, 5% in the capillaries, and 65% in the veins. Why? Because the veins have a large lumen. So more volume is circulating at a certain point in time. Now, as we look at the different types of blood cells that we have red blood cells, and we have platelets, and we have white blood cells. And there are two types of white blood cells, lymphocytes and phagocytes. But basically, they all start from the stem cell, which is found in the bone marrow. And then, of course, they differentiate into the different cells. So there's the myeloid stem cell and the lymphoid stem cell, and then they develop into red blood cells, platelets, and white blood cells. Now, the red blood cells, what are their functions? The red blood cells basically contain hemoglobin, and they are only for the transport of oxygen. So they form oxyhemoglobin, and oxyhemoglobin is an unstable compound. So when it reaches the tissues, then the oxygen is given up. So it doesn't hold on to the oxygen tightly, but when in the tissues, when the oxygen partial pressure is low, so the oxygen is given up by the red blood cells. So red blood cells basically are only one purpose, and that is to transport oxygen. Now, the white blood cells are of two types. One are the lymphocytes, and these lymphocytes produce antibodies. So you must be able to pick up the lymphocytes because you have to label them in different micrographs, and you have to pick them up in diagrams. So they produce antibodies. While this one is a phagocyte, and a phagocyte actually does phagocytosis, which you will study in the next chapter, which is on immunity. So phagocytes engulf, digest, and destroy pathogens. Engulf, digest, and destroy pathogens. While platelets are what? They're cell fragments, and they're only responsible for the clotting of blood. Now, what we have to understand is clotting of blood is very important. Why is it very important? Because if a person cuts himself, or if he, by mistake, the knife cuts his skin and he starts to bleed, well, he will bleed to death. So basically, what happens when the blood clots is going to prevent the excessive loss of blood. I said, look, if the blood clots, it's going to prevent excessive loss of blood. And also, because now it is a cut surface, so it is going to, air contains a lot of bacteria, viruses, pathogens. So all those can reach the interior of the body. So it's just like your skin is it's a beautiful protective layer on your body, which prevents any pathogens from entering your body tissues, which are just below the skin. And below the skin, of course, you have your muscles and you have your bones and you have all the organs of the body. So the skin is the most beautiful protective suit that you could wear. Just like, in space, they wear these suits. So basically, your skin is that suit which you wear, which is going to prevent the entry of pathogens, and that is going to make it it's the best protective suit that you could wear. So the platelets are there. You see, when you cut yourself or you damage your skin, and you start to bleed, so the platelets release an enzyme, and that results in the fibrinogen being converted to fibrin, and that forms a clot. And that, of course, is the main mechanism of platelets. Now, again, another diagram. So I want you to pick up this, the lymphocyte. This is also called the neutrophil, or it's also called the phagocyte. And of course, this is the red blood cell. And these are the platelets, which I just said were cell fragments. So these are the basic ones which I want you to understand. I want you to pick them up very easily. So this neutrophil is also called a phagocyte. Now, coming to the functions of the plasma, and what is a plasma? Plasma, as I said earlier also, it's a liquid part of the blood. You see, the blood is composed of two things. Number one is the cells, and number two is the liquid part. The liquid part is just like we have aloo gosht ka shorba. Now, aloo and gosht and the shorba, which is the liquid part. So, aloo and gosht would be the cells in it, but the shorba or the liquid part is called the plasma. So transports what? What is the function of the plasma? You must know that. There's a very interesting MCQ on this. So transports, number one, the red blood cells, the red blood cells and the white blood cells and the platelets. So three of them, the RBCs, the red blood cell, the white blood cells, which are of two types, and the platelets, which are cell fragments, as I said earlier as well. So the blood cell is a separate entity. Then we have ions, sodium ions, potassium ions, chloride ions, sodium chloride. Then glucose, which is soluble, and all the rice and the roti and the fries that you had, all that is digested and ultimately ends up as glucose in your plasma, in your blood plasma. The word that we use is blood plasma. And then the amino acids. So if you had meat, or if you had fish or chicken or an omelette, all that protein is digested to amino acid, and that enters again your blood plasma. Then we have the hormones being transported, insulin being produced in the pancreas and being transported to the liver where it works. Then we have carbon dioxide is transported as bicarbonate ions. Then the urea, which is formed by deamination in the liver, is transported in the blood plasma. Remember this, when you write it, you must write it correctly. You say urea in the blood, and I don't give you any marks for that. And then you argue with me and say, oh, well, miss, I should get a mark. And I say, no, but you didn't. You just said blood. You didn't say blood plasma. And then, of course, we have the famous plasma proteins, which are large molecules, but they're soluble molecules. Please remember, nothing insoluble can be transported in the plasma. So all these are soluble products, but you have to know them in these different headings. Number one, blood cells. Number two, ions. Number three, glucose. Number four, amino acids. Number five, hormones. Number six, carbon dioxide, urea, and plasma proteins. So these are the headings which I want you to remember, these headings, and pause the video here and have write down your notes and remember these. So if we just number them for your own little purpose, 1, 2, 3, 4, 5, 6, 7, and 8. But then there's a ninth one as well. And the ninth one is vitamins. Now, when you have orange juice, well, you have vitamin C in it. When you have milk, you have vitamin D in it. So all these vitamins don't need to be digested, and they'll be absorbed directly into the bloodstream. But where are they carried? They're not carried by the red blood cells or the white blood cells. They're carried in the blood plasma as vitamin C, as vitamin D, as vitamin B. Of course, we don't study B and all, but we study C and D only. So the vitamins are also another component which is part of the blood plasma. Now, this is another very important graph which I want you to look at. And basically, this understands this is the area, this is the velocity, and this is the pressure. Now you can see how the pressure is decreasing. This is the aorta, pressure is the highest. You can feel the pulse in that, arteries, then arterioles, then capillaries, then venules, veins, and the vena cava. So you can see how the pressure is decreasing and then it remains less in the vena cavas. But let's look at the velocity. The velocity is high here, then in the arterioles it decreases, and then in the capillaries it's less, and then it again returns, but it doesn't return to the same level as the arterioles. But in the vena cavas, the velocity is slightly more than what it was in the capillaries. Now, this is the capillaries here. Please understand that. Now, the area, what do we mean area? Cross-sectional area. So, the cross-sectional area is very little in the aorta, then in the arterial arteries, then arterioles, then the cross-sectional area starts to increase. Then the cross-sectional area is the highest in the capillaries. This is the capillaries here. So the cross-sectional area is the highest in the capillaries, and then it slowly comes to decrease, and then of course in the vena cava it is about the same as in the aorta. So this is the difference that we have to understand as how the pressure, velocity, and the cross-sectional area changes as we go from one blood vessel to the other. Another graph showing you this, and we can see how the three different graphs are shown here differently. So velocity, blood pressure, and total area, which is the cross-sectional area in centimeters square. Now, the next topic which we need to understand is how this tissue fluid is being formed and why is it being formed. So there is I always like discussing it is as the arterial end of the capillary. You can see this is the arterial end of a capillary. Now here the pressure is very high. So a lot of stuff is going to move out. And on the venous end of the capillary, then things are going to come back in. So the pressure is very high, 10 millimeters of mercury. Here it is minus 7 millimeters of mercury. You don't have these figures, but you have to understand is that there is an arterial end of the capillary. The pressure is high. So everything is going to move out. Water is going to move out. Glucose is going to move out. Amino acids dissolved in it are going to move out. So all this stuff, when it comes out, once it was inside, it was called plasma. Once it comes out, it is called tissue fluid. So as soon as this fluid is out, it is not called plasma, it is called tissue fluid. And we have the body cells outside, which is the tissue fluid is surrounding these body cells. But then at the venous end of the capillary things, the water and the dissolved substances are going to move back in. But some of this fluid is not going to move back in, and that is going to enter the lymphatic system. So the lymphatic system will be the one which the tissue fluid which is unable to return, that tissue fluid is going to enter the lymphatic system. Now basically to understand this, I just draw an imaginary line on a capillary and I say, well, this is the arterial end of the capillary and this is the venous end of the capillary. No, so a lot of pressure is being exerted at the arterial end of the capillary and all this is moving out. So the plasma is moving out. Once it moves out, then it is not called plasma, it is called tissue fluid. So the tissue fluid surrounds the body cells. The tissue fluid surrounds the body cells, and these are the body cells which I've shown you in red. So the tissue fluid surrounds the body cells, and because of high pressure, all this is going to leak out, like water is going to come out, then the glucose is going to enter the cell, the amino acids are going to be used by the cells, and this is going to be basically more diffusion into the body cells. But then you have to understand is why is water entering back into on the venous end of the capillary? Why is all these fluid coming back into it? There's a very important reason for that. As I told you, the plasma transports plasma proteins. These are large soluble molecules. Now the plasma proteins cannot move out. They cannot move out. Why? Because they're large molecules. So they stay inside the capillary. But then what has happened? Water has moved out on the arterial end, so there is less water here. Now this makes it a very concentrated solution. So when this makes it a concentrated solution, so now water will move in from a higher water potential to a lower water potential into the venous end of the capillary. So basically it is the plasma proteins which do not move out cause the blood in the venous end to become more concentrated, because now the plasma proteins are the same. So if there were 12 here or 6 here, there's 6 still there. They haven't moved out of they can't move out because they're large molecules. They cannot move out of the small pores which are found in the capillary. So plasma proteins remain the same, but then there is less water here now because the water moved out here at the arterial end. So there's less water, so now a lower water potential is generated and water starts coming back into the venous end of the capillary. So this is what you have to understand, the exchange of substances at the capillaries, how it is different at the arterial end and how it is different at the venous end. And this only can happen in the capillaries. Why? Because arteries and veins, of course, have thick walls. So nothing can move into and out of the vessels in the arteries and the capillaries. Now I want to just discuss a little bit about phagocytosis. What happens? How does phagocytes, what do they do? Sometimes you say, miss, they eat bacteria. Well, that's wrong. They don't eat bacteria. They don't have any teeth. So bacteria becomes attached to membranes, evaginations called pseudopodia. It sort of develops these false. Pseudopodia means false arms. You can see these false arms developing. And the word we use is engulfs. And then we say bacterium is ingested, forming a phagosome. Then phagosome fuses with lysosomes. What are lysosomes? They contain hydrolytic enzymes. And they're little structures inside the cell which have been made by the Golgi body, which actually help in, they contain pockets of, the little bags of enzymes. And these bags of enzymes are going to attach to this and then actually going to digest that bacteria, because the bacterial cell wall is made up of a protein, peptidoglycan. So these enzymes, which could be a protease, which could be a lipase, will digest the wall and the membrane. So the phagosome fuses with the lysosomes, and lysosomes enzyme digests the captured material, and the digestion products are released from the cell. So phagocytosis: engulf, ingest, digest, and then of course the products are released from the cell. Exocytosis. Another very interesting one, it says entrapment, then formation of food vacuole, then fusion with lysosome, and then digestion. Digestion means it's actually destroyed. But you can't say the word biologically correct is digestion, because it's the enzymes which are going to digest it. Just like we digest food in our intestines, like you have rice, so the starch is digested to maltose and then to glucose. So the word digestion has to be very clearly understood. What do we mean by digestion? Large molecules into, and then of course we talk of physical digestion and chemical digestion, which we've done in the chapter on human nutrition. Another diagram for you to visualize it very clearly: entrapment, formation of phagosome, degradation, and then exocytosis. So just to mentally visualize it, that how this cell is going to get hold of the bacteria and then destroy it, which is happening inside your body. So these are your these are your protectors. They are going around in your body, and whenever such a foreign bacteria or something enters, which is a foreign material, which is not your own body cell, so the phagocytes recognize that these are not my own body cells. Okay, let me destroy them. These are strangers, and they could cause harm to your body. So a beautiful way of protecting your body against invading aliens, just like it's a science fiction movie. And the last topic which I want to cover is lymphocytes. What do the lymphocytes do? They produce antibodies. So you have an invading pathogen like a virus or a bacteria. And what do the lymphocytes do? They produce antibodies. And antibodies lock on to this antigen. Antigens are the surface markings of the virus. We've done this in next chapter, which is the chapter 12 on immunity. So please go on to that video and have a I've done a detailed account of that in that video. So the antibodies actually attach onto the virus or the pathogen, and then the phagocytes come, and the phagocytes are the big policemen, and they come and they ingest and digest the invading pathogen. So antibodies are produced by lymphocytes. Lymphocytes produce these antibodies, and the antibodies attach to the surface of the invading pathogen, and then of course it destroys it. So this completes your second video, and thank you very much for watching, and thank you once again. Completes the chapter 11, and this is the second video on this chapter. Thank you very much.